Welcome to all of you for this Hyderabad Lecture Series. And I request the Dean of the School of Life Sciences to accompany with the Honorable Vice Chancellor and the guest of the day, Professor Erwin Neher. May I ask them to come to the dais, please? The Vice Chancellor to take the proceedings.
Good morning, everybody. Welcome you back for this Hyderabad Lecture Series at University of Hyderabad. We are very happy that we are able to have the second lecture by another Nobel Laureate today. We had a wonderful talk yesterday, and we have our distinguished speaker to talk to us this morning. I would like to first extend a warm welcome to our speaker, Professor Neher, for accepting our invitation at a short notice and also making it possible to visit Hyderabad and then deliver this lecture at University of Hyderabad under Hyderabad Lecture Series. I also would like to thank Professor Eva Maria. She's another distinguished scientist accompanying her husband, but in her own capacity, she is a highly distinguished scientist and doing wonderful things at Gottingen. I extend a warm welcome to you also, Professor Eva Maria. As I told yesterday, this particular Hyderabad Lecture Series is meant for disseminating the knowledge and inviting distinguished people to deliver lectures for the benefit of the community at Hyderabad. As part of these lectures, now we are having this third Nobel Laureate speaking to us. We have people from different institutions who have come here. Different universities have come here, and we have officials from IRDAI, that is Insurance Regulatory and Development Authority of India, who have been the sponsors of this event in a way by setting up an endowment. They are also here. So we have a diversity of audience, diversity of cutting across disciplines. People have come from different places, and it is not possible for me to extend welcome to each and every group separately. Therefore, I take this opportunity to extend a warm welcome to everyone who is here with us in this room and in the neighboring room, and also those who are watching this live telecast, live streaming of this lecture, Hyderabad Lecture Series. I welcome everyone for this lecture. I also want to again emphasize and inform every one of you that this has become possible for us to have the distinguished speaker with us because of the Hyderabad Lecture Endowment that we have at the University of Hyderabad. This endowment was set up in 2013 by Insurance Regulatory and Development Authority of India, and we all thank IRDAI for sponsoring this, for setting up this endowment and facilitating this kind of interaction and facilitating the opportunity, giving an opportunity for all of us in Hyderabad and also those who are watching us from elsewhere to have this great opportunity for all of us. Let us thank them once again. I would not like to take more time, but it is also important for me to inform that this university has been a highly reputed university in the country, and we have always been in the top ranking institutions in the country. And we are among the few institutions which figure in the world ranking. Therefore, we always take pride that University of Hyderabad has always achieved excellence so far, and in fact we are now moving towards eminence. As part of this, I should also make a mention, without taking too long, just one point I want to mention, that in an exercise that we have done recently about the contributions made by University of Hyderabad faculty, particularly in research, we have realized close to 300,000 times the research work done by University of Hyderabad faculty has been cited in research. This is a remarkable achievement for any university in the country, and we are proud of it. And we are now proud to have a Nobel Laureate who is going to speak to us. I don't want to stand between you and our distinguished speaker, but before that, only a few minutes I would like to request Dean School of Life Sciences. Very few minutes, sir. I don't want too long about the School of Life Sciences because it is a general lecture that we are eagerly waiting to listen to Professor Neher. Please, just a couple of minutes.
So let me join the Vice Chancellor in extending you a warm welcome to this Hyderabad Lecture Series on behalf of the School of Life Sciences. It is indeed a great pleasure to honor to have the distinguished speaker, the Nobel Laureate, Professor Erwin Neher, and Mrs. Neher with us, and to deliver the distinguished lecture under this Hyderabad Lecture Series. The School of Life Sciences, as our Vice Chancellor was pointing out, is one of the largest in the country. The faculty working in divergent areas of biology and biotechnology, and we contribute to the growth of this university in terms of the research, both having national and international interactions with various institutions and also with the industry. It is indeed a great pleasure to have this distinguished lecture to host this lecture in the School of Life Sciences. I welcome you all again. Thank you.
All of us require, of course, may not require an introduction, but it is customary to have an introduction of a distinguished speaker, for which I will be requesting Professor Ramaya from Department of Biochemistry to introduce the speaker to all of us. Before that, I want to make one important point. While University of Hyderabad is hosting, and IRDAI has been the supporter, has set up the endowment for this lecture series, this particular lecture series has become possible. Yesterday and today's lecture, as I mentioned yesterday, has become possible because of one person, that is Professor Mrutyunjay Saur, who has been behind and who has worked as a faculty of University of Hyderabad. He has helped us to organize this event at University of Hyderabad. Thank you, Mrutyunjay. Professor Ramaya to introduce the guest.
Thank you very much, sir. Honorable Vice Chancellor, Professor Apparao Garu, Madam Eva Maria, and Professor Erwin Neher, the distinguished German guests, the distinguished academicians and industry people coming from various academic and research institutions and industry from Hyderabad, deans, heads and faculty of the various schools at the University of Hyderabad, Professor Mrutyunjay Saur, the director of Kalinga Institute of Industrial Technology, who is instrumental in bringing the German guests to the University of Hyderabad, students, ladies and gentlemen. I welcome you all to this Hyderabad lecture series organized by the University of Hyderabad with the help of IRDAI funds. And today we have a distinguished speaker with us, Professor Erwin Neher, and I am really honored to introduce him. It is indeed a great honor and privilege bestowed on me to introduce the speaker, and I hope I'll do the justice. Professor Neher was born on March 20th, 1944 in Landsberg, Bavaria, and raised in Buchloe, 70 kilometers west of Munich, with physics background and influenced by cybernetics and Hodgkin-Huxley theory of nerve excitation. During school education, Dr. Neher went to study biophysics at the University of Wisconsin at Madison on a Fulbright scholarship in 1966. His master's project, directed by Professor Bevan, was an early attempt at producing molecular beams of macromolecules for mass spectrometry. In 1967, he returned to Munich and obtained his Doctor Rerum Naturalium in physics from the Institute of Technology, Munich. Subsequently, his quest to do research in biophysics in an area related to nerve excitation is realized when he joined with Dr. H.D. Lux at the Max Planck Institute for Psychiatry. Here, he worked on voltage clamping in snail neurons and with single ion channel recording in artificial membranes. To circumvent space clamp problems, he used suction pipettes for local measurement of current density. At Dr. Lux's laboratory, he met Dr. Bud Sackman, who had experience in the neuromuscular junction. Neher and Sackman collaborated, aiming at the measurement of single ion channel currents, which involved in developing and refining the patch clamp technique. This technique allows researchers to measure individual ion channels in the cell membrane to the level of picoamperes by eliminating the membrane's electrical noise. The classical paper, actually, which is titled Improved Patch Clamp Techniques for High Resolution Current Recording from Cells and Cell-Free Membrane patches, which is published in 1981 in Archives of European Journal, has received 18,823 citations as on date. Until then, they were using a pipette 1,000th of a millimeter in diameter fitted with an electrode to detect the flow of ions through a single channel in the cell membranes. Patch clamp technique allows the measurement of incredibly small electrical currents amounting to a picoampere that pass through a single ion channel. These studies revealed the regulation of these ion channels for the movement of positive and negative ions, showed how the ion channels function, and has contributed to the understanding of the cellular mechanisms underlying several diseases, including diabetes and cystic fibrosis. The technique, which records how a single channel molecule alters its shape and controls the flow of current within a time frame of a few millionth of a second, is a path-breaking research in the area of ion channels that led them to bag the Nobel Prize in 1991 in Physiology and Medicine. Of course, after 1983, Dr. Neher's interest also shifted away from the channels themselves to processes they initiate inside the cells, eventually leading to a cellular response like secretion of hormones and neurotransmitters. Professor Neher held several posts and obtained several laurels, and some of them which are coveted here are listed, and I'm speaking now about them. He was a postdoctoral researcher in the laboratory of Dr. Stevens' laboratory at Yale University, Department of Physiology, New Haven, Connecticut. He is a Fairchild Scholar, California Institute of Technology, Pasadena, USA, in 1989, and served as the director of the Membrane Biophysics Department at the Max Planck Institute for Biophysical Chemistry, Gottingen, Germany, during 1983 to 2011. He won several awards, and the most coveted among them include the Gottfried Wilhelm Leibniz Prize, the highest honor awarded in German research, Louisa Gross Horowitz Prize from Columbia University, Feldberg Award from Feldberg Foundation, Fidia Research Award lecture from Fidia Research Foundation, Harold Lamport Award from the New York Academy of Sciences, Spencer Award from Columbia University, Geithner Award from Toronto 1989, and Bristol Myers Squibb Research Award from New York, Gerard Prize, American Neuroscience Association. He was conferred also honorary doctorates from Dr. H.C. Lumberg University Centrum in 1988, from the University of Pavia in 2000, and honorary professor at the University of Gottingen in 1986. He was elected foreign member of the Royal Society in 1994. Madam Professor Eva Maria is also a very distinguished scientist in the areas of biochemistry and microbiology. She obtained her diploma in biology in 1974, earning a PhD on biochemistry from the University of Gottingen. Her thesis was regulation of the biosynthesis of polybeta hydroxybutyrate in alkaligens eutrophs H16. She then worked in the same university as a staff scientist in 77. From 77 to 78, she also undertook postdoctoral research at the Max Planck Institute for Biophysical Chemistry in the Molecular Biology Department of Gottingen. While at the Charles Stevens Laboratory at Yale University for postdoctoral work, Dr. Maria met Professor Erwin Neher, and she worked in Neher's Young Investigations Lab and married him on 26 December 78. She was a staff scientist at the Institute for Physiological Chemistry at the George August University. She also taught experimental courses in chemistry and biology at the Free Waldorf School, Gottingen. Later in 2000, she founded XLab, an experimental laboratory for the youth bridging the gap between high school and university. She has been its managing and executive director since 2004. I welcome both of them, and ion channels are the key mediators of external chemistry and internal biology. In the context, whether it is chemistry or biology that is important between people to bring them closer or away from each other, I would say ion channels are a kind of bridge between ambience and cellular and molecular signaling. It is very fascinating to determine how the chemistry of the environment influences the physics or generation of electrical signals, which in turn influence biology. This is how biology, which started as a hobby, has grown into an interdisciplinary science. With these few words, I would welcome once again the guests.
I now request Professor Erwin Neher to deliver his talk.
Can you hear me? Yes. Yeah. Okay. Well, dear Professor Ramaya, dear Mr. Vice Chancellor, students, colleagues, ladies and gentlemen, it's a real pleasure to be here to give a lecture as part of this distinguished series, and it's a pleasure to learn about the research on one of the top universities of this country. So my topic, ion channels, is something which fascinated me from high school days, as Professor Ramaya already pointed out. In order to convey a little bit of this fascination, let me show you a few historical slides to start with. Okay. And of course, talking about the history, now I think you can also hear me from this microphone. Okay, so talk about the history of bioelectricity. That has to start with the famous, one second, yeah, with Luigi Galvani and Alessandro Volta, two famous Italian scientists who more than 200 years ago found out that you can stimulate nerve of a frog and make it to switch, which was a phenomenon which was very exciting for people at this time, and which started lots of investigations into what was considered to be bioelectricity. Now, 100 years later, the famous Spanish neuroanatomist Ramón y Cajal showed us in his beautiful drawings that the nervous system is a filigrane network of what we now call neurons. Today we know that our brain is a network of about 10 to the 12th of such neurons, which are connected with each other by synapses. Each neuron, on average, receiving input from 10,000 other ones. Ramón y Cajal already had a good idea about signal flow in the central nervous systems. If you look closely at his drawings, you see in many places these little arrows, which actually indicate what he thought was the flow of signals. And we must say today that although all his ideas originated just from looking through the microscope at dead and fixed tissue, he was mostly right. Of course, he didn't know at this time what these signals were. But around the same time, a physiologist in Berlin, Julius Bernstein, built this machine and recorded for the first time very short-lived signals, which we now know are so-called action potentials or nerve impulses. He recorded these at the cut end of nerve and muscle, and could show that there are these electrical changes which happen on the millisecond timescale or sub-millisecond timescale. He also developed what's called his membrane theory, namely the idea that the electrical signals originate at the membrane of neurons of cells, which surrounds every cell in a tight way, so that it separates electrically the inside of the cell relative to the outside. And his theory stated that the electrical signals you see and which propagate through the nervous system are actually generated at this membrane. This was a very important concept for subsequent work. So 50 years later, in 1952, British physiologists Hodgkin and Huxley published their famous work about the action potential, showing that it actually is a electrical wave which propagates along the nerve fibers being a hollow cylinder of membrane. And they were able to reach these conclusions by working on the squid, which was known to have a giant nerve fiber, a membranous cylinder of about almost 1 millimeter thick, where they could insert wires longitudinally into it and record currents flowing through the membrane upon applying a voltage to the membrane. And they could show that applying such voltages elicited currents which were carried by sodium ions and by potassium ions. And they separated these two components by ionic substitutions and could show that the sodium currents were short surges of inward current upon depolarization, whereas potassium currents were switched on in a delayed way but persisted. And then casting these time courses into differential equations, they could actually demonstrate that a membrane with these properties, separating a conductor inside from a conductor outside, would generate electrical waves which propagate along the axon, along this cylinder of membrane. So this was the state of knowledge when I got interested in it, and as a student, wanting to do a PhD project, entered the laboratory of Dieter Lux, a very well-known electrophysiologist. So what you see here is actually a recording from my own thesis, which I think illustrates beautifully this concept of electrical excitability as worked out by Hodgkin-Huxley. Namely, in my thesis, I worked with conventional microelectrodes, very small glass pipettes pushed into the soma of cells. In the case of my thesis, it was giant neurons of snails, a garden snail. So I recorded the potential inside relative to outside, which is at rest, as I'm sure you know from basic electrophysiology, has a resting potential of about -60, -70 millivolts. If you then depolarize this cell by injecting positive current, you get a slowly developing positive change in voltage, which after stop of the excitation drops back to basal levels. However, if you surpass a certain threshold, then this phenomena sets in, described by Hodgkin-Huxley, namely due to the depolarization, a surge of inward current into the a cell which makes it positive, followed by a delayed outward current. Though this phenomenon at that time was understood on the macroscopic level, as a consequence of changes of permeability changes. However, the big question at this time was, what is the molecular mechanism of these permeability changes? There were many ideas in the literature. Of course, the idea of Hodgkin-Huxley that there are gates which switch currents, which suggest that there might be pores in the membrane which actually open and close. But there were competing ideas. It was known that these permeability changes are very specific for ions, certain ions, sodium, potassium. And the only mechanisms known with such high specificity were carrier-mediated currents, such as currents mediated by valinomycin, a very specific carrier for potassium, which carries 1 potassium ions at a time across the membrane. There were ideas that there may be phase changes in the membrane, in the lipids, And we discussed these ideas in detail. And at one time, Bert Sackmann, who was doing his PhD at the same time in the same institute, and myself decided that we wanted to prove the original Hodgkin-Huxley concept of pores by just recording current with sufficiently high resolution that we would be able to demonstrate step-like changes in current when these channels, these pores, open and close, which was, of course, expected to happen in a discrete manner. But there was a problem. From some circumstantial evidence, we knew that the currents to be expected should be in the picoamp range, in the range of 10 to the minus 12 ampere. Now, to give you an idea about this order of magnitude, there is this arrow which is supposed to represent the quantity current in logarithmic terms. And you know that typically currents flowing in the light bulb are around an ampere. However, currents in electronic circuits are typically only 1,000, a milliamp, But these are also currents which you can find in living systems, but only in very special ones like the squid giant axon. Typical currents in electrical tissue are still ordered by 3 orders of magnitude, the microamp range. These are currents as they are elicited by the nerve in the muscle, when the nerve signals to the muscles that it should contract, elicited by the opening of the neuromuscular transmitter acetylcholine. We know that these currents are made up of so-called miniature currents, which are currents flowing in response to a single transmitter-containing vesicle being releasing its transmitter, its acetylcholine, towards the muscle membrane. And now we know that these currents, again, are made up of single-channel currents in the picoamp range 1,000 times smaller, 10 to the minus 12. Now, the problem was that all methods available for recording electrical currents in biological tissue had an intrinsic noise of a few tenths of a nanoamp, 100 times larger than the picoamp current which we wanted to measure. So we had to come up with a new idea, with a new technique, and this resulted what's now known to be the patch clamp technique. Unfortunately, some panels shifted here. It's written down below, patch clamp technique. The name comes from the fact that the technique consists, or the basic idea of the technique is not to push microelectrodes into the cell, but to place a microelectrode, a microglass pipette, touchingly onto the surface of the cell, trying to insulate a little patch of membrane for the electrical measurement, with the underlying idea that in case there is an ion channel, a pore switching on this little patch, opening and closing, if we apply a voltage across there, connect this whole assembly to a amplifier, we should be able to record these tiny currents. And actually, after just 2 years or so of experimentation, we arrived at a state where we could first see these kind of step-like changes. Now, this is not electrical excitability. This is currents elicited by the neuromuscular transmitter acetylcholine, which was contained in the pipette in this experiment, opening so-called acetylcholine-activated currents. Now, the key to the success was recognizing that, as you can find in electronics textbooks, that each electrical measurements is confounded by the so-called transistor noise, or just a noise resulting from the fact that electronic charges, being electrons or ions, are quantal in nature. This leads to so-called Johnson noise or resistor noise, which is given by this equation. And you can see that the resistor noise is the smaller, the larger the resistor or the impedance of the signal source. So just solving this equation for how high a signal impedance you need in order to have, say, current recording on the picoamp scale, you find, say, putting in a bandwidth of a kilohertz or so, that the impedance of the signal source has to be hundreds of megohms or higher. And the problem is that by just placing a pipette onto a cell, you never get such high resistance, because there is always a kind of a aqueous leak between the pipette and the membrane. So the big problem was to prevent this leak and to increase the signal impedance to the point that you reach hundreds of megohms of seal. So finally, we succeeded in another step forward in around 1980 to achieve very high steel resistances in the gigaohm range, and this allowed us to then record these current changes in very good time resolution. Now, as I mentioned, these first recordings were obtained on the neuromuscular junction, on the membrane of frog muscle cells. At the same time, biochemistry had advanced to the point that the so-called acetylcholine receptor could be extracted by biochemists as a membrane protein, characterized as a membrane protein. It turned out that the acetylcholine receptor is a pentameric membrane protein with binding sites for acetylcholine, and obviously what happens when acetylcholine binds to this receptor, then the protein undergoes a conformational change, opening an ionic pathway to let ions enter the cell. So these kind of records more or less were the first records where actually one could watch a biological macromolecule do conformational changes in real time. Now, with the improved resolution, we could also return to the original question, namely of electrical excitability, what's the basis of Hodgkin and Huxley, and this was done together with Fred Sigvors, who joined the lab as a postdoc. He did an experiment on another type of muscle which has high electrical excitability and could show that when depolarizing this muscle, he could see upon repeated trials, repeated sweeps, these inward blips of current happening primarily shortly after switching on the electrical stimulation. And taking the ensemble average of many of such currents, he could show that actually the mean current was a transient inward current, just the same as Hodgkin-Huxley had shown originally on the squid giant axon. Now with these two types of measurements, the proof was established that both electrical and chemical excitability is mediated by this discretely switching ion channels. Okay, so this was what we aimed at and what we achieved around 1980. What comes in the rest of my lecture is actually not our own work, but the work by hundreds of laboratories worldwide who adopted our techniques and came up with a number of surprises about ion channels. The first surprise was that ion channels are actually not limited to what's called electrically excitable tissue, like nerve, muscle, some kind of neuroendocrine cells, but that a huge number of different types of ion channels fulfills various tasks in different cell types. So a number of them are listed here, but this is just an excerpt. I could produce a list with hundreds of entries. Okay, the second, well, just to give you an example of the kind of multitude of different ion channels, I have here this example from the regulation of the heartbeat. In the heart, in heart cardiomyocytes, you have action potentials, somewhat like nerve action potentials. However, here in the ventricle of the heart, you have an action potential with a very broad shoulder in order to allow the heart, give the heart enough time to contract. And as in the nerve, the upstroke of this action potential is carried by sodium ions, but what's shown here is a number of potassium currents, different types of potassium channels, which contribute to the fine regulation of the shape of this heart action potential. And you can see there are listed here 10 different kinds of potassium channels. And in the ventricle, these 4 types listed here contribute potassium currents with their specific time courses. Different regulation is in the atrium, where these currents contribute relatively little, but another collection of different types of potassium channels makes the atrial action potential to be finely regulated also by hormonal influences. Okay, so the next surprise was that ion channels are prime targets for pharmaca. This actually was not so big a surprise, because there had been empirical knowledge accumulated about so-called calcium antagonists, substances which antagonize the inflow of calcium in the heart and also in the blood vessels. And this is an example of the action of one of these calcium antagonists, nitrendipine. It's a slide given to me by Harald Reuter, a pharmacologist from Bern, Switzerland, who was very lucky working on cardiomyocytes that in some cases he actually had a patch with just one channel of a very particular type, namely a calcium, a voltage-dependent channel carrying calcium current. And when he depolarized this patch of membrane, then he noticed that this channel just flickered open and close in very rapid succession. This was in control. Under the influence of this calcium antagonist nitrendipine, openings were much more rare. The current was much more the channel was much more reluctant to open, so that you had only occasional little blips, and correspondingly, on average, a very much smaller inflow of calcium, reducing the contraction power of the cardiomyocytes, and also reducing the tone on the blood vessels, because such channels also occur in the endothelial cells of the blood vessels. Just a few more examples of drugs acting on channels. One is memantine, which is the second most prescribed drug for ameliorating the symptoms of Alzheimer's disease. It is not a cure for Alzheimer's disease, but patients taking it somehow can better cope with the consequences of Alzheimer's disease. And it is turned out to be a blocker of NMDA-type glutamate channels. Glutamate channels are one of the major excitatory channels in the brain, in the synapses of the brain. And this substance blocks one specific subtype of such glutamate channels. Another example is VX77, a compound which was found to be an activator of a transporter protein for chloride, which is dysfunctional in certain forms of cystic fibrosis. And this turned into an FDA-approved drug for this disease, which is very helpful. Unfortunately, only for a very small fraction of patients who have specific mutations in this channel, and that's why it's called a genotype-specific medicine. So this brings me to the third surprise, namely that ion channels are the causes, or defects in ion channels, mutations in ion channels are the causes for a number of hereditary diseases. Again, a few of them are listed here, but as in the case of drug targets, I could list hundreds of different diseases. This is not surprising because almost 200 of our 30,000 genes code for ion channels, and well, I'll come back to that. Let me say a few more words on this specific case of the cardiology regulation. Now there is this cardiac syndrome of long QT syndrome, which means a prolonged plateau phase of the cardiac action potential. And this clinical syndrome can be caused by very different dysfunctions of ion channels. And this is quite clear when understanding the mechanism here of this prolonged plateau phase of action potentials. As I mentioned already, there are many different types of channels contributing. There is a short-lived sodium current. There is a longer-lived calcium current, which maintains the plateau at the same time provides for the influx of calcium into the cardiomyocyte. And then at some point, when potassium channels are switching on, the balance between influx and outflux tips, and you get this repolarization. Now the time point when this happens can be shifted in two ways. It can be either prolonged by a gain of function in the influx pathway, and this has been shown in certain patients, that there is mutation in the sodium current which prolongs, which conveys to the sodium current a long tail, which then leads to delayed repolarization. But the same phenomenon can also obtained by a loss of function mutation, namely if the counteracting potassium currents are smaller. And indeed, both of these phenomena can be observed from cardiology. There are 12 congenital types of this syndrome known, which clinically all look very similar, but as I pointed out, the mechanisms underlying these different types are very different. So, due to the prominent role of ion channel regulation in cardiac physiology, and due to the experience that many drug candidate molecules which work out perfectly well in animal models fail in clinical studies due to arrhythmias in the heart and other cardiology problems, there has been an early FDA ruling that all candidate drug molecules for any target have to be tested against possible side effects on one of these potassium channels in the heart, namely the so-called HERC channel, which seems to be a sponge, act like a sponge, attracting, binding all kinds of molecules which would otherwise would be very suitable for as drug candidates, but then fail because they act on this channel. More recently, a new initiative by the FDA has been started preparing protocols for extensive testing of drugs for their influence on the cardiomyocytes. And this now includes 5 different types of ion channels which need to be tested, will be needed to be tested in the future before FDA will approve for clinical tests and finally, of course, for the substance itself. Now, after this, let me finish with a somewhat amusing role, I think, of ion channels. And this refers to thermal, our sense of heat, of warmth. And you know, probably from physiology again, that in the skin of your there are many different types of receptor organs, nerve endings, specialized structures, and so on. Among them, ion channels localized in nerve endings, which open and close in response to temperature changes. And there are, in fact, quite a few of such channels, all of them from a family of channels which have not been known before the advent of patch clamp. It required the very sensitive methods to measure currents in order to find these kind of background channels which act in the resting state, you know, and they're all from this so-called TRP family. TRP stands for transient resistor, transient receptor potential. They were first characterized as parts of the light transduction pathway in Drosophila vision. So a number of these are temperature sensitive. Some of them act in the temperature range of the very cold between, so this, for instance, this TRPA1 switches on and off between 0 and 10 degrees. On the other end of the spectrum, there are channels which open only with noxious heat, but together they cover the whole range of what we call physiological temperatures. Now, the surprise came when researchers found out that these channels not only open in response to temperature changes, but that also certain substances can influence their open-close behavior. And no surprise, if you look what are these substances, you find that capsaicin, the ingredient of hot pepper, activates this TRPV1. On the other end of the spectrum, menthol, the ingredient of mint, opens TRPA1. And from this insight, of course, it's clear why you feel hot or cold when you eat, or why you feel hot when you eat a spicy Indian food. So with this, let me close, but not without mentioning once more that all the second half of my lecture was basically reporting on what happened to the field with the advent of the new technique. But the original studies, of course, were done together with Bert Sackmann, with whom I shared the Nobel Prize. And most of the very exciting developments in the early 1980s, when it turned out that there are so many different types of ion channels that you could do many more things manipulating channels, not just recording channels. And this was done with a number of postdocs who had joined our labs. First of all, Fred Sigworth, who did the electrical excitable cells. Alain Marty, who invented what's called the so-called whole cell, tight seal whole cell technique, a technique to get access to the cell interior after rupturing of the patch, which turned out to be very important. Owen Hamel, now a professor at Galveston, who found out that you could remove patches from cells so that you could study ion channels in isolation with good access of active compounds from the inside and the outside. So all of this then was a collaborative effort, and I'm happy to acknowledge the contributions of these people. Thank you for your attention.
Let us put our hands together to thank Professor for this wonderful lecture now. He has agreed to address a few questions. Maybe I will leave it to the floor. First opportunity to the guests and next opportunity to the insiders.
Professor, I'm a physician. I have a great interest in pharmacology throughout my career. And thank you for the great work and being here in Hyderabad. My question to you is, in the current era of drug development that is happening, purely rooted in molecular biology, and we all understand the current day work, which is rooted on the inflammatory mediators, whether you take TNF alpha, interleukins, and all of those things. If perhaps in history, if these were discovered back in the times when you're working on the ion channels, do you think that the entire magnitude and the science of ion channels would have been submerged? Or my other way of understanding is, what is the relationship of the ion channels to the new discoveries in molecular biology? And how do you see the future drug development?
Yeah. Well, I think the future of drug development will consist of two flows, you know. One is the flow of information about individual targets, drug targets, and the other flow is how then components isolated acting on individual drug targets act together, you know, to give a formulation of medicine which goes beyond the single component medicine. And of course we have many examples how modern research can contribute to finding new drugs. I mean, the example I showed here on the cystic fibrosis transmembrane regulator is I would say, success story, which started by, or one contribution of this was the isolation of the characterization of the cystic fibrosis transmembrane regulator as a protein which has very complex properties, you know, ATP binding, being either a transporter for chloride or a channel for chloride, very complicated physiology, and then finding molecules which boost the function of this transmembrane regulator in cases there are certain mutations which deteriorate its normal action, you know. And I can imagine by just finding drug targets, finding molecules which act on it, being able to characterize the molecular action, being able then to see how dysfunctions, restored functions on the molecular level turn into functional changes on the cellular level, and finally translating into clinical symptoms that will tell us a lot about human biology, you see. Particularly recent work on isogenic cell lines derived from human patients, you know, where you can have cell lines carrying the disease mutation, and then you, by site-directed mutagenesis, you correct this. Now you have two cell lines, so-called isogenic cell lines, one carrying the disease, the other one not. You can compare things from on this exactly the same genetic background, diseased versus normal, on the molecular level, on the cellular level, on the clinical outcome. And there are hundreds of different genes, channeled genes, known to be mutated in certain diseases, you know. For each of, say, a clinically defined hereditary disease, there are, for many of these, there are 10, 20, 30, 40, 50 different mutations known, leading to clinically similar but slightly different outcomes. I think studying this whole wealth of possible information there on human cell lines will tell us a lot about human biology, which in the end will give much better predictions for possible clinical outcomes of drug candidate molecules than animal models, you know.
Dr. Mohan Rao here, first row.
Thank you very much for an exciting talk. Almost all the cells in our body have these ion channels, as you mentioned, and they influence almost all the body functions, including thinking and digestion and things like that. Ion movement happens under the field of electric field, and that you talked about. What is your opinion about the effect of magnetic field on ionic channels and current. If you apply high magnetic field, would the ion channels start functioning differently?
No, I'm not aware of any work. Of course, I mean, you would expect from physical grounds that sufficiently high magnetic fields should influence any current, you know. But given the length scale, you know, of flow across channels, it seems that just the orders of magnitude needed for such effects are not reached in normal life. You know, even people going into the magnetic resonance scanners, you know, experiencing very high fields, don't have cognitive consequences. You know,
we don't know if they have consequences because we were keeping them only for a brief period, so we don't know if there are any consequences.
Well, of course, there may be long-lasting consequences, but I think from at least from cellular studies, there is no evidence about that, you know, and also from purely physics consideration, I think the field strength required would be much higher, you know, than what's currently reached. Of course, there are, say, certain phenomena, say the localization of birds, you know, is known to be dependent on magnetic field, but this is mediated probably by very specialized proteins which contain metal centers or so, which are special for sensing magnetic fields.
Yes. Antesha.
I thank University of Hyderabad for giving this great opportunity to us. And thank you so much for your lecture, sir. My small question is, where do your great ideas come from?
Pardon? What?
Where do your great ideas come from?
Where do you get these great ideas is the question.
I thought I explained this, you know, that the Hodgkin-Huxley was kind of inspiration, you know, and I'm not sure whether it was such a great idea to put a glass pipette onto the surface. I think we were just very lucky that it worked, you know, and we were very lucky that once it worked so well, it turned out that there are so many different types of channels. But I think that one of the prerequisites to have great ideas, to find solutions, is a very elaborate curiosity, you know, which makes you keep thinking about your problem, trying different approaches, trying to look at the given problem from all possible sides, you know, and just keep on. I mean, we didn't see our currents the first time we tried, but it required some kind of persistence. It required some kind of thinking about what's the basis of the failure, why just placing the pipette onto the cell doesn't immediately show channels, that you need this high, this very high quality seal, very high quality contact between the pipette and the membrane, and working towards this goal makes you finally succeed.
Thank you, sir. Thank you, sir.
Yes.
I'm Professor of Physiology. It was a great lecture which we teach to undergraduate students also, on the patch clamp technique. They're very excited about it. My question is, have you found any relevance in malignancy, in cancer, the ion channels, anything going wrong haywire?
Well, I have not, you know. I mean, I left the field of ion channels about 20 years ago and specialized on synaptic transmission, release of transmitters. But colleagues of mine worked in the cancer field and found out many links. You know, on the one hand, there is this one type of cardiac channels which was studied by my friend Walter Stümer, who showed that one of these channels, which is expressed normally in the heart, if it's expressed ectopically in other tissues, it creates tumors, you know, and so the turned out to be a good diagnostic tool, expression of this channel in non-cardiac tissue as a marker for tumors. Then a number of colleagues are looking at the role of certain calcium influx channels, ICRAC, and some of these TRP channels as with respect to their roles in cell cycle regulation, and in some cancers, the expression of these channels is either increased or decreased, you know, and so there is definitely a role of channels in cancer, but I think this is just due to the fact that cancers develop all kinds of tricks to overcome the immune defense, and so you can find mutations in any signaling part of the genome, you know, including channels.
Nice, thank you.
Good afternoon. Welcome to Hyderabad. My question is there any crosstalk between the GPCR modifiers into the ion channel modifiers?
Yeah.
In drug discovery, at least.
What modifiers?
The G protein coupled receptors, agonist and antagonist. Do they have any crosstalk with the ion channel modifiers?
What kind of modifiers?
Agonist or antagonist, any one of them.
Yeah. Well, I mean, there are many GPCRs which act like neurotransmitter receptors. The so-called metabotropic glutamate receptors are receptors for the neurotransmitter glutamate, you know, which then triggers, I mean, the binding of glutamate to these metabotropic channels triggers whole cascades of intracellular signaling, say for also the muscarinic cholinergic receptors. There are 5 subtypes, you know, so you can find any kind of second messenger pathway being influenced by one or the other of these metabotropic glutamate receptors or muscarinic, and there are many more.
Can I ask another one? May I ask just last one? Are there any, the way you predicted the thermal and the chemical induced?
Keep the microphone closer.
Oh yeah. Are there any chemical, since you mentioned already about the chemical and thermal variations in these ion channels, are there any light induced ion channels that modify their activity? Light induced.
Light induced. Well, I mean, naturally there are not so many, but in recent years, this family of so-called channel rhodopsins was discovered, which are rhodopsins which react to light, but the reaction will be the opening of a channel. And this turned into this very popular field of so-called optogenetics. You know, people now express these light-activated channels in various parts of the brain, everywhere in the tissue, so to control nervous activity by light. And this is area of research which is expanding rapidly, and contributes a lot to understanding of.
Thank you very much.
There are a couple of hands on the back.
Professor, I just have a small question to a big problem. We all are aware of the multi-drug resistance problem. So most of the microorganisms, they're effluxing out, like efflux pumps are involved in the multi-drug resistance. So do you have out of that, now we are more than 50 years in the field, so do you have a best solution for this problem?
For multi-drug resistance? No. No. I mean, I've not worked on this, and multi-drug resistance is probably not a phenomenon which relates to a single kind of signal, but just to a combination of mutations and things which knocks out a number of possible drug targets, but I've never worked on this field and have no specific knowledge on it.
Thank you for the nice lecture. I just need a comment. All the ion channels are for cations. How do you think about anion related ion channels?
Yeah. No. I mean, there are many anion specific channels known. I mean, the original work on the electrical excitability was sodium, potassium, calcium, cations. But subsequently, the list of channels discovered which carry anions is very long. I mean, the example I showed about the cystic fibrosis transmembrane regulator is a chloride transporter or channel, depending on the conditions. Many of the inhibitory neuro transmitters are opening chloride channels. GABA, for instance, glycine is opening chloride channels to hyperpolarize the membrane.
Thank you.
One girl wanted to ask, give her a chance first.
Yeah.
Thank you for wonderful talk. Actually, I have some doubt about. Actually, my topic is different. You are talking about natural proteins. What is your opinion about synthetic ion channels in drug delivery?
Synthetic ion channels in drug delivery.
In drug delivery. Synthetic ion channels, which type?
Any means for cation or anion, any kind of synthetic ion channel, just
Yeah.
like carbon nanotube, anything, just like cyclodextrin.
Yeah. Well, I'm a little bit skeptical about the carbon nanotubes. I mean, there has been work published in prominent journals about recording with carbon nanotubes, but for me these experiments have never been very convincing. Now, synthetic pores, of course, play a big role in nanopore sequencing, but I'm not so much aware of roles of synthetic channels in drug delivery. So,
Thank you.
There was one more. Somebody raised a hand there.
No.
No?
No.
No more?
Okay.
Professor Ramaya.
Sir, are there there must be cell-specific channels and also general channels.
Also?
General
channels, yeah.
And my question is now whether the general channels, are they different from cell type to cell type in their functioning?
I see.
Or they have a conserved structure and function?
No, of course, I mean, it depends on the cell type, which of the 200 channels in the channel genes are expressed, And there are certainly a number of housekeeping channels which are involved in cell volume regulation, which all cells need, you know. But I don't think there is some kind of a rule, you know. But this is similar to.
But these ones which are maintaining, I mean, which are ion channels maintaining the basic cell functions, whether their efficiency of functioning, would it be dependent on the ambience, or
I see.
whether it is regulated by some small changes in their structure and function?
Well, I mean, there is the example of the so-called piezo channels, which are channels which regulate or which sense membrane stress, you know, and these channels have been shown to be basically expressed everywhere, you know, in each cells. So it's just an example, and on the other hand, of course, there are very specific channels like the voltage-dependent sodium channels, which are restricted to excitable tissue, you know. But as I said, I don't think there is kind of a rule.
Okay.
Sir, one last question.
Yes,
Last question?
I think that one boy want to ask something.
So I was waiting.
Sir, thank you, sir, for the lecture. So as you mentioned, many diseases are there due to defect in ion channels. So have you studied different diseases in plants? So I'm from plant biology, that's why I'm asking.
I see.
So have you studied in different diseases and different effect of drugs on that ion channels?
Well, for sure, I haven't studied disease channels in plants. And I must say, I have done work on plants recently with together with a colleague, Erwin Neher from Würzburg. So I have been in contact with plant cell research, but I'm not aware of the specific link between channels and plant diseases, you know. I would just have to go into the literature and look around. I'm sure in the vast literature on regulation of ion channels and all these phosphorylation mechanisms which regulate ion channels in plant cells, that there must be cases relating to plant diseases. So, but
thank you, sir.
So, sir, as you said, when you discovered it, it was quite unanticipated that these ion channels had a important effect on the were very important for the drug use, or important for drugs. So, having made this discovery, how far do you think that there's a moral responsibility on the scientists like you and other, that how your discovery is being used, and how far do you find that over a long journey you have been successful on controlling how your discovery has been put in the real practical use?
Yeah. Well, of course, I mean, scientists are responsible for what they do, the consequence of their work. They are responsible for using the funds which are provided by society in a good way. But of course, scientists are not alone. There is a huge community of scientists, and there is kind of job sharing between scientists. And I think that my special abilities are more on the side of making, of creating basic knowledge, you know, and of course any new basic knowledge should be checked for possible applications, you know. But as I said, I'm not alone. I hope that other scientists take up whatever we find out in new basic knowledge. And I mean, as the example of ion channel research shows, I mean, we did our work 30 years ago, which led to this breakthrough in measurement technology. And 30 years later, there are plenty of practical consequences, practical applications. But it took a long time until this translation of basic advances into practical things. And of course, there is always this debate between should we do basic research, should we do applied research? I think the answer is we should do both, and be sure that there is the right mix between the two, because basic knowledge creates basic research creates new knowledge, and it's always easier to find applications for new knowledge relative to applications for old knowledge, because old knowledge has been looked at already by so many people from different angles, that you must be very lucky to find something real new. However, if you have new knowledge, your chances are much better.
Okay, I think we close the questions here, and let us have a pleasant duty to felicitate our speaker. For this felicitation, I would like to request two more people to join me on the dais. One is Shri Satheji from IRDAI, and also Professor Eva Maria onto the dais, please. I now request Professor Naresh Babu to present vote of thanks.
On behalf of University of Hyderabad and School of Life Sciences, we would like to thank Professor Erwin Neher for accepting our invitation and giving an inspiring and thought-provoking speech on ion channels. He delivered a brief history of ion channels and ionopathies and the importance of ion channels in cardiomyopathies and cardiac functions. And also, in questioning, he also said students and research scholars they should think about the problem and surrounding the curiosity to solve the problems. That is most important to solve any biological problem or any problem related to the biological sciences. And also, I would like to thank IRDAI for Insurance Regulatory and Development Authority for setting up a grant corpus fund for organizing this kind of Hyderabad Lecture Series, and also accepting to organize this event at a very short notice. Thank you, sir. And we would like to thank Professor Apparao Padale, and is one of the Vice Chancellor, is untiring efforts to bring the university to at eminent status, and by bringing eminent scholars like Professor Erwin Neher to University of Hyderabad as a part of Hyderabad Lecture Series. Thank you, sir. And we would like to thank Dean, our School of Life Sciences. He is always in forefront in organizing this kind of events, and is very enthusiastic to organizing this kind of events, and we are very happy to have Dean, and thank you, sir, for coordinating this event. And this event wouldn't have been possible without the help of Professor Mrutyunjay Saur, Kalinga Institute of Industrial Technology, for bringing this Erwin Neher to this university and delivering this very important lecture on ion channels. Thank you, sir. And we would like to thank coordinator Professor K.V. Ramaya, Madhubabu, and other student volunteers who went along couple of days ago, we have a very short notice, and they went through the cabs and delivered the flyers to all institutes across the city. And we would like to thank the student volunteers who helped in organizing this event. Thank you. And we would like to thank PRO's office and Dean's office and other members who helped in organizing this event, and in particular Pranav, who able to live streaming this event to show this event across the university. Thank you, sir. I hope I thanked everyone. If I did, and I would like to thank the distinguished guests from outside of the university and other institutes, and also faculty colleagues and students for making this event a grand success. Thank you, one and all.